Hello everyone, my name is Artem Isaev. I recently defended a PhD in Skoltech, and I'm working in this lab for more than 4 years. And so now we are in the laboratory of molecular biology in the Center of Life Sciences, and we carry multiple projects here. My group studies interaction of phages with their bacterial hosts. Also, we study here CRISPR-Cas defense systems, and we carry a large project that is devoted to the metagenomics of Arctic oceans. Our laboratory is equipped to carry different types of projects. So first of all, we are growing bacterial and viral cultures. Then we also run sequencing in this laboratory, prepare a library for Illumina sequencing. The usual workflow might include detection of the gene of interest, its cloning, and next we can follow with protein purification, and it is performed on this kind of very efficient system. So we run chromatography, and then we can study in vitro activities of proteins of interest, and also we can study protein-protein interactions, and for this we have a biolayer interferometry octet system. So now we can go to the main part of our laboratory where we carry the majority of work with phages and bacteria. This is the main part of our laboratory, and open space when each student have their own individual tables, and here we carry most of microbiology, cloning, and biochemistry types of work. So one of the main directions of our studies is novel phage defense systems. And this is an area of research that is bursting with new discoveries. So 10 years ago, we knew about restriction modification and CRISPR-Cas systems. But nowadays, every month, new papers appear in Nature and Science that describe novel types of phage defense systems. So we are trying to catch up with this new wave of novel systems, and we also study their mechanism of action. And in turn, phages or viruses have evolved to inhibit these host defense systems to survive. And also we study viral anti-restriction proteins, so this intricacy of interactions between phages and their hosts. And we have a lot of projects devoted to this field of phage-host interactions, and we are waiting for new students to join our lab and carry this fascinating research.
So, hi everyone, I'm Masha Tutukina, and I'm a senior research scientist in the lab of Mikhail Gelfand. So what actually we're studying? We're studying mainly bacterial transcription, and from both fundamental and practical sides, let's say. Initially, we work on phylogenetics, and we predict something, for example, a binding site for a transcription factor, and then we studied it experimentally to prove or vice versa reject our predictions. And what we're doing for this, mainly we're doing ChIP-seq analysis and sequencing, and after this sequencing, we definitely need to check our results with the qPCR. And here we have the qPCR machine that helps us in doing this. Also, we can test some samples via electrophoresis. And in order to understand if a transcription factor does something to a bacterial growth, we use this reader, which permits us to measure the growth curve even if we are at home, for example, sitting watching a film. This reader will work for us and will monitor our culture growth and show us the growth curve on the monitor of this computer. And if, for example, bacteria stop growing in the absence of one or other gene, this means that this gene is essential for the growth in these conditions. And we then continue studying this effect, study RNA-seq, or ChIP-seq, or qPCR, or making mutant on, for example, transcription factor, or vice versa, overproduce it. To overproduce proteins, we use these lovely shakers, the small one and the big one, for big flasks, up to 1 liter. And then we centrifuge our cells and go to the chromatography system and purify our proteins that we then can use to produce antibodies in the rabbits and make ChIP-seq, for example, analysis. So this is one part of our work in the lab. And another big part of our work is connected to metagenomics. We, for example, take a soil sample or sample from our nose, and I'm trying to understand what microbes live there and how they're connected to the, for example, our health, let's say. Or in the case of soil studies, we're trying to understand how, for example, some pollution affects the composition of microorganisms in this or that soil. And how we do this? First, we isolate the DNA from our cell samples using, for example, these centrifuges. We homogenize the sample and using the special kits, like this one, very lovely, pure linkings. We isolate DNA, and then we go to the PCR machines. So we then go to the PCR machines. There we amplify the variable regions of 16S ribosomal RNA, which allows us to then to make phylogeny of bacteria. So we put our samples here. Then the machine does the work for us. Then we purify the amplicons and send them to the sequencing facility of Skoltech. And they sequence our samples, and then we analyze them and reconstruct the phylogenetic composition of bacteria and even metabolic pathways, if we are lucky. So, for example, one of our transcriptional projects is dedicated to alternative way of lactose degradation. So we all know that bacteria have just one way of lactose degradation, described by Jacob and Monod in 1950s, Nobel Prize. But we, using our some phylogenetics approach and comparative genomics, we noticed that the way of degrading of sulfoglucose in our E. coli is very similar to a way of lactose degradation in bacilli. And now Anya and Margarita are studying exactly this pathway of sulfoglucose degradation in E. coli, and I'm trying to understand if it indeed can utilize lactose. So even in E. coli, we can have something new, and it's still a very interesting object of study. It helps us a lot. We're waiting for new students. We'll be very happy to host them in this lovely lab to study new phagers or new RNA polymerases or new metagenomics samples and new transcription factors in bacteria. And please join Skoltech.